Uh, a very good morning to you, ladies and gentlemen. I would like to welcome you to the fourth day of our SAAB 22 conference, which is actually the final day of the conference. I hope we enjoy the day as we used to do from Monday up to here. It was an enjoyable week, so I hope we can close it in good style. Before we start, I've got two short announcements to make, then we can move on. The first one is about the photo competition, which I said something about yesterday when we were closing. The competition has been activated. So at the moment, all the photos have been uploaded on the website. So we request you to go to the website. When you get the website, you go to photo competition. After photo competition, you go to vote. When you get there, you choose your best photo, then that will be fine. So that at the end of the day, we can actually choose the one that would have been voted most. So the competition opens now and it will close at 1 o'clock. So make sure that if you want to vote, you do that between now and 1 p.m. Then the second announcement is for those who have been doing adjudication from Monday up to yesterday, but mostly those who did Tuesday and Wednesday. We still have some forms that haven't been submitted by now. So we request you to submit those forms as soon as possible so that we can keep on pushing our program smoothly. So please, those who are doing adjudication, submit your adjudication forms as soon as possible. Thank you very much. So we are now going to kickstart our last day of the conference with a plenary lecture from Dr. Longara, who is based at the University of the Free State, Kwakwa campus. Dr. Ngara completed an MSc in biotechnology at the University of Zimbabwe in 2000 before enrolling for doctoral studies at the University of the Western Cape in 2005. She obtained a doctoral degree in 2009 before working in the same university as a postdoctoral research fellow and lecturer on a fixed-term contract. In September 2013, Dr. Ngara joined the University of Free State as a lecturer in the Department of Plant Sciences at Kwakwa campus. She currently holds a senior lecturer position and supervises postgraduate students in a range of projects in plant stress biology. From 2016 to 2020, Dr. Ngara held a Royal Society Newton Advanced Fellowship hosted by the Department of Biosciences at Durham University in the United Kingdom. Generally, Dr. Ngara's research interests lie in understanding how crops, particularly cereals, respond and adapt to a range of abiotic stress factors such as drought, salinity, heat, either individually or as a combined stress factors. The line of research is motivated by the need to develop crops that produce maximally under harsh environmental conditions and thus enhancing food security. With the current projection in the global climatic change and its effects on food security, the need to increase crop production under hot and dry conditions is increasingly becoming a priority worldwide. So Dr. Ngara's small but growing research group uses system biology approaches to answer such questions. So today, Dr. Ngara will present a topic to us as a plenary lecture entitled Sorghum Responses to Abiotic Stresses, fitting one puzzle piece at a time. So, Dr. Ngara, I welcome you. Thank you very much for accepting our invitation to come and present your talk to us. And I wish you the best in your presentation and also in the conference. So, colleagues, I hand over to Dr. Ngara to kickstart a plenary lecture. Thank you very much.
Thank you. I'll just share my presentation with you. Please let me know if you can see my presentation on screen and if you can hear me clearly. Is everything fine, Jackie?
Very well. So you can proceed.
Okay. Good morning to you. I'm going to speak to you about some of the work we are doing on sorghum responses to abiotic stresses as we try to fit one puzzle piece at a time. Agricultural productivity is vulnerable to droughts, and crop species as well as farm animals die due to heat stroke as well as inadequate water supply when these drought episodes are severe. The picture on the on the right, I took it from a newspaper article I read in November 2015, which was describing some of the devastating effects of the drought episode South Africa experienced during that growth season. In this particular picture, cows were looking for water to drink, but the dams were drying out. So this particular calf was looking for water in a drying dam in Ladysmith. The water was just in small patches in the center of the dam. The calf got stuck in the mud. It couldn't free itself and it died. So our research focus focuses on understanding how plants respond and adapt to a range of abiotic stresses, including drought, heat, and salt. And in our experiments, we are using sorghum, which is a cereal crop, the fifth most important cereal crop in terms of global production. It grows well in hot and dry environments, and it is used for food, feed, and biofuel. But why are we using sorghum plants? We view it as a potentially good model system for these plant stress biology studies, mainly because it is naturally tolerant to drought. It is a wide genetic diversity and its genome has been sequenced, thus providing an important resource tool for molecular studies. In our studies, we're mainly using sorghum seedlings under lab conditions, as well as sorghum cell suspension cultures, mainly when we look at secreted proteins, because my other interest is on what lurks in the extracellular matrix of cells, of plants. So in 2014, Prof. Ndimba and I published this review article on model systems in drought stress proteomic studies, and we're giving our perspective on Arabidopsis and sorghum. In that article, we also highlighted the importance of conducting comparative studies using stress-tolerant versus susceptible sorghum lines as we try to shed more light on how sorghum adapts to the dry and hot conditions. How far have we gone? We've generated quite a number of proteomic data sets from tissue-specific proteomic data sets from whole plants, but we've also generated quite a number of proteomic data sets of secreted proteins when we were using cell suspension cultures and for different experiments. For the sake of today's talk, I'm just going to briefly tell you about one of the drought studies we had and even in the heat stress project, which is still ongoing. So the sorghum drought project was largely driven by Tatenda Goche, who was a PhD student. So we received a large collection of sorghum seeds from Dr. Nemera, our collaborator from the ARC Grain Crops Institute. And when he provided the seeds, he gave us their drought phenotype. So some of them were drought susceptible, some of them were tolerant, which was good for the set of experiments we were hoping to conduct. So Tatenda eventually worked with ICSB 338, which is susceptible to drought, and SA 1441, which is tolerant to drought. And his experiments were in a soil potting experiments in the lab, incubating growth chambers rather than greenhouses, because we didn't have the facilities then. And drought was imposed by withholding water for a number of days, so like 8 days for his main experiments. And then we measured a number of physiological and growth parameters, osmolyte content. We performed a root protein analysis using eye-track technology, and we also validated some of those differentially expressed proteins using QPCR. So one of the studies, the first experiments we did was just to, check what was happening to these two sorghum varieties before we really proceeded with much of the experiments. Although we knew one was susceptible to drought, one was drought tolerant, we grew them in soil pots. The pots were about 22 centimeters in depth. After 1 week, within the first week, we were well watering them. So you can see that in panel A, well watered plants, ICSB 338 and SA 1441. After the first week of well watering, we just didn't water them anymore. We just left them in a collaborator's lab in the greenhouse, and this is what we saw 2 weeks later. The soil was drying. 338 plants dried out much faster than SA 1441. As you can see in the close-up pictures of C and D. So yes, for some reason, these plants had different drought phenotypic traits, which was good. So we moved on to measure a range of parameters using another experimental system that Tatenda was working on here in Kwakwa. And in this one, we measured the relative water content, which really assesses the water status of a plant. And we understand that the leaf relative water content is related to a large number of other physiological variables: leaf growth, stomatal conductance, photosynthesis, transpiration, and so forth. In these experiments, Tatenda had 3 treatment groups: the controls, which were well watered throughout the experiment. What is written there in black says water limitation, which were basically the drought-stressed seedlings for 8 days, or 8 days with no watering. And what is labeled there as recovery were just the drought-stressed plants plus 1 day of re-watering. For the sake of the presentation, let's just look at the controls and the drought-stressed plants and forget about the recovery one. So we observed a decline in relative water content in both varieties with drought stress compared to their respective controls, but the decline was more pronounced in ICSB 338. So, for some reason, maybe SA 1441 was able to conserve its water. We also measured the chlorophyll content of these plants during the 8-day drought stress period and 24 hours later when we re-watered them. And for the chlorophyll content, we observed a decline in a sharp decline in chlorophyll content for the drought susceptible line as early as day 4 of the treatment. But for the drought tolerant variety, it sort of, delayed a reduction in chlorophyll content up until about day 7. For some reason, maybe it was able to protect its cellular machinery. With stomatal conductance and leaf surface temperature, these parameters were measured simultaneously using a leaf parameter, and we observed, we observed a decline in stomatal conductance with the progression of this treatment time, whilst the leaf surface temperature gave us an opposite a response, which is really an increase in leaf surface temperature. And makes sense because when the stomata are closed, there's no free ventilation, so there is an increase in leaf surface temperature. Then we also measured root and shoot length and biomass readings, but I'll only concentrate on the root and shoot length, the first 2 graphs at the top of the slide. Graph A indicated that following drought stress, both varieties had increased root length. But in B, following drought stress, the ICSB 338, which is susceptible to drought, responded by declining the shoot length. However, the tolerant variety, which is SA1441, the length of the shoots did not change following that particular drought stress treatment period. So in the collaborators lab in the UK, Tatenda then conducted a time course experiment, once again imposing drought by withholding water and harvesting leaf and root samples for various analysis. For osmolyte content analysis, root proteomic analysis, as well as the gene expression analysis. During the course of the study, Tatenda would say he observed leaf rolling as the drought stress was progressing, wilting, in some cases drying out, and death in one of the varieties. So the next task was to measure osmolyte content. So we understand that plants accumulate compatible osmolytes, various types, in response to abiotic stresses. And these osmolytes serve protective functions, stabilizing the structure and function of biomolecules within the cell. However, the different types and amounts of osmolytes may vary between and within plant species, the type and severity of the stresses, and in some cases differ with plant tissue. So here we measured, this was measured proline and glycine betaine using some Hillick-based procedure in our collaborators lab, and we had samples during the course of the drought stress treatment from 0 to 12 days, and we observed a general increase in both proline and glycine betaine in the roots of both tissues during the course of the drought stress treatment. However, SA1441, which is drought tolerant, seemed to accumulate larger amounts of these osmolytes, especially at day 8. We also observed that the roots had much more glycine betaine than proline. Once again, in the leaves, both osmolytes were identified. There's an increase in accumulation as the drought stress period progresses in both varieties. Once again, SA1441 seems to be accumulating both of those at higher levels than the susceptible variety, except of course for proline at 12 days. And once again, here we observed that proline content 12 days following stress was much higher in leaves than proline was much higher in the leaves than glycine betaine. So there was some, tissue-specific accumulation of osmolytes there, but there was also varietal differences in the accumulation of those. But both varieties do accumulate these for a range of functions during drought stress. Okay. So our main objective in this particular experiment was to study the sorghum root proteome in response to drought. What is happening within the roots during drought stress? And we did this bit by using initially a 2D Dodge gel-based proteomic approach, and we used that primarily for quality checks before we went on to quantitative analysis using the iTrack method, which is non-gel based. So in the collaborators lab, once again, in 2D Dodge, what happens is that we would have replicate samples for the controls of each variety and their corresponding drought stress treatments. We can label those with specific dyes, maybe a red one for controls, a green one for the drought stress treatments. We perform 2D gel electrophoresis with those samples individually in their own gel casts and so forth. After that, we scan with a specialized scanner and software, and we are able to quantify the changes in expression. But for 2D Dutch, we would overlay the gels, a control gel with a drought gel, to give you what we call gel overlays at the bottom of the screen there. So what do those colors mean in the gel overlay? So if you're to see any red intense color in the gel overlay, it means that protein spot, or what I didn't say is the spots that you see in the gels just represent a protein spot which was separated in 2 dimensions. So a red spot in a gel overlay means that the quantity of that particular spot was much higher in the control sample. So it was that one that was downregulated during drought stress. A green spot in a gel overlay would be a protein spot which had high levels in the drought treated sample compared to the control. So it was those that were upregulated. But any of those yellow spots means that there was equal amount of that particular spot in the control and the drought. So there was not any differential expression in that particular spot. So because this was just a quality check, we could see that something was happening. There were proteins that were being expressed, and there was some differential expression within and between each of those varieties. Then we moved on to iTrack, which is a non-gel based technique. In iTrack, you would have, we started off with 4 biological replicates for each treatment group per sorghum variety, labeled them with appropriate tags. There are different tags for each sample, and then mix them together and subject that to LC-MS. At the end of the day, you get raw iTrack data, which would sift through and clean up. After the cleaning up, we ended up with 1,169 positively identified root proteins from ICSB 338, which is susceptible to drought. And of those, over 1,000 proteins, 234 of them were differentially expressed in response to drought, 47 were upregulated, and the rest were downregulated. So we use bioinformatic tools to get to know, which molecular, we did the geo analysis. We also had to find out if there are any conserved domains in some proteins and family names, because most of them came out as uncharacterized. So we really didn't know what they were. So this table just shows you one of the 19 pages that he produced for ICSB 338. We did the same for SA1441. Just over 1,000 proteins were positively identified after the cleaning up exercise. 184 of those were differentially expressed, 61 upregulated, and the rest downregulated. So for the sake of this presentation, I'm not going to be looking at the individual proteins because this was a global proteomic analysis, but I'm just going to try and look at the putative functional groups, and then we see if we can draw a story out of that. So if we're to look at all those differentially expressed proteins between the 2 sorghum varieties, we found out that we could actually categorize them into some putative functional groups such as ROS detoxification, metabolism, transcription, protein synthesis, and so forth. But some of them still remained unclassified because we couldn't get much information about them online, in the online databases. Nevertheless, further interrogation of this data showed us that we had about 51 proteins that were common between both sorghum varieties, and some of them were upregulated, and these include the ROS detoxification related proteins and the signaling proteins, and the downregulated ones were mainly metabolism related proteins. However, we also had unique proteins, as we saw in those Venn diagrams. In ICSB 338, 186 of those proteins were unique to the susceptible variety. but there was this marked up-regulation of proteases and a marked down-regulation of protein synthesis, as well as metabolism-related proteins. On the other hand, in SA1441, out of the 136 unique proteins, we found a marked downregulation of proteases. So the proteases were really, downregulated. But amongst the upregulated proteins were protease inhibitors, protein synthesis related proteins, transcription, as well as ROS detoxification and signaling. So we could see this difference just by looking at the putative functional groupings of the proteins, in which were up and down regulated between the two varieties, looking at the unique proteins. And of course, the question would want to know why was this happening? Well, in our thoughts, we would say that the drought susceptible variety, which is ICSB 338, which I've written in green there, reduces metabolic activity and protein synthesis, and also increases protein degradation under water stress or drought stress, possibly as an energy-saving mechanism to support cell processes that are really critical for plant survival. But what about the other variety, which is tolerant to drought? We would suppose that, the higher expression or the upregulation of all those proteins, transcription, protein synthesis, detoxification, signaling proteins, and so on, is a way of increasing a pool of regulatory and stress responsive proteins that generate effective protective mechanisms against the prevailing stress. It was just our thoughts then, but I know there's quite a lot that is happening in these plants. So this work was then published in 2020. You can read more about that work there, but mainly the proteomics data set and a bit of the physiological data. Nevertheless, we were left with more questions than answers. For example, we were asking ourselves, really, what is the significance of such a differential protein expression pattern between those two different varieties exposed to drought stress? Shouldn't we be looking at functional studies, or shouldn't we be mining the data more to see what is really happening, especially more for the uncharacterized proteins, which we just label as unclassified? But maybe is there something that we can learn from those unknowns? The next question was, do other cereal crops display similar trends between drought-tolerant and susceptible varieties, such as wheat and rice? And we're working on these, and the wheat study is more or less at an advanced stage, but we don't have those answers yet. But also, I was also keen on looking at root phenotyping, because I see the root as an important, well, every tissue is important, but I'm saying in order to sense water deficits, the root should be playing a major role in those processes. There are a lot of signaling related processes that are occurring there. We also want to do root phenotyping, but for this we would need systems that allow unrestricted root growth, unlike what we're currently using, and also merge that with the phytohormone and signaling system analysis and see what is happening in the roots and specifically in the leaves. The other question was also to investigate the transcriptome changes both in time and space between those two varieties. And we already started this project last year with our collaborators at the ARC biotechnology platform. So that was the drought story, just a brief summary. So I just want to, let me just see the time. I just want to go move quickly on the heat story, which was run by 3 master's students, and they did different parts, contributed to the work in different ways. So Elelwane began by establishing colors from ICSB 338 I know she had 8 sorghum varieties she started off with, one of which was white sorghum, which was used by Dr. Ndimba and I earlier in the 2000s. And then we also had SA1441. The plan was, okay, maybe we can have one line which is tolerant, the other one which is susceptible, and we can have these 2 cell lines, but it didn't work according to plan. She could only get friable colors from ICSB 338, and all the tolerant lines just wouldn't budge in terms of colors induction, but that was fine. So she did experiments with other work, but the cells she generated and colors were then used by Mamosa, was investigating the proteomic changes of secreted proteins of these cell cultures. But in her work, she also did a bit of cell metabolic activity readings, osmolyte content analysis, and also analyzed the expression of marker genes following heat stress prior to the proteomic analysis. So the sorghum cells we have, even the colors, we are maintaining it at about 27 degrees Celsius, plus or minus 2 degrees. And they grow well. So she started off her heat stress at 35, then we didn't see much change in cell viability, then she went on to 40. Yeah, so in the graph A on your left shows the cell viability readings of ICSV 338, which was exposed to 40-degree heat stress for 72 hours. Yes, there was a reduction in viability at 48 and 72 hours, but it wasn't really that huge. Then obviously, yeah, we weren't sure really if this was good enough for us to conduct a proteomic analysis with such a mild decrease. So in a collaborator's lab, they have Arabidopsis thaliana cells. So they said, okay, let's just check if this 40-degree treatment of ours is actually working. So Arabidopsis is really heat liable. It doesn't like heat at all. They grow it normally at 22 degrees. So a 40-degree heat stress was really shocking to the cells. At 72 hours, they were more or less dead. So yeah, 40 degrees was working, but Arabidopsis and sorghum 338 were responding differently, which was good. Then Mamosa also went on to check the expression of 2 marker genes, heat marker genes, HSP 70 and 90, just to see if there was something happening at least at the molecular level. And she found out that yes, there was something happening. They were sensing the heat, and there was an increase in both those marker genes with time under heat stress, which was good. Then she measured osmolytes, because we have read that, you know, these osmolytes, there's a range of them, but proline and glycine butane are important in stabilizing biomolecules under a range of abiotic stresses. So we said, okay, let's just measure it while she's still in the collaborator's lab. Then she did it. The proline results, well, the proline even at 0 hours was very low, lower than what we had observed previously in the leaves and so forth. And then at 72 hours, there was a decline in proline content. I thought maybe it was going to go up, you know, since with an increase in stress, then you have increase in proline, but it didn't work out. So I asked Mamosa, you know, did we not mix up the sample? He says, no, this is what it is. And then with the glycine butane results, you know, we couldn't detect anything then. So it was like, oh, Mamosa, what happened? But there was nothing there. Then obviously these results, we haven't really taken them into the public domain because I just wasn't sure of what was happening then. But the rest of the proteomic work was then published for Mamosa in 2020. When Samgeliswe came in, she was looking at combined stresses using ICSB 338 once again, doing a lot of other things. But then I said, let's look at these osmolytes once again, because there's something that I don't understand. But the question is, how did we determine the combination of stresses to use? Well, for Samgelisiwe, the combined stress was a combination of osmotic stress and heat stress. Osmotic stress was induced by 400 millimolar sorbitol. that we had previously used and published with Elelwane in the scientific reports, and this had been used by other researchers as well, especially in Arabidopsis. The heat stress was at 40 degrees as determined by Mamosas experiments. The combined stresses were 400 millimolar sorbitol and with incubation at 40 degrees, whilst the control samples were maintained at 27 with no sorbitol. I also want to say those osmotic stress cells, it was just exogenous addition of sorbitol, but being maintained at 27 degrees Celsius. I'll just show you the proline results from her study. I don't have the stats analysis here, but they were done. What it shows is, okay, so it was over a 72 period, so would sample cells just before we started the experiments at 24 hours, 48, and 72. At 0 hours, the samples control, osmotic stress, heat stress, and combined stress samples all had more or less the same amount of proline. That was before the stress. But 72 hours after the stresses had been applied, then we found a massive accumulation of proline in osmotic stress cells, but a decline, statistically significant decline in the heat stress cells, but an increase in the combined stresses, although it's not as high as osmotic stress cells. So in fact, the decline that we observed in Mamosas experiments, although the quantities were low even at 0 degrees, is actually what is happening. Heat stress suppresses the accumulation of proline in sorghum ICSB 338 cells. Then we still didn't know why. I'm not showing the glycine betaine here time-wise, results here, but when we looked at glycine betaine at 0 hours, it was like 50 times less than that of proline. And the content did not vary much between the time points from 0 up until 72. It was like nothing is happening there except for a decline that we saw for heat stressed cells at 48 hours. So obviously, you know, glycine betaine doesn't seem to be playing much of a role in these cells if they are heat stressed. So in last year, we published a review when we're just looking at what's happening in whole plants of sorghum, transcriptome and proteome-wise in response to drought stress. And there we illustrated this diagram, which just gives us, you know, a flow of information relating to the transcripts and proteins that we could actually gather from various papers, those relating upon perception of stress. You know, there's stress signaling, there's gene expression and regulation, and there is accumulation of a whole host of stress responsive proteins, including compatible osmolytes. So we found transcriptomes of biosynthetic enzymes of a range of these particular osmolytes. And we also learned that some sorghum drought responsive transcriptomes are reporting beta-alanine betaine biosynthetic enzymes. And we found out that this beta-alanine betaine is known to accumulate in stress-tolerant plant families. I think this is on shrubs and so on, where it possibly serves as an alternative osmoprotectant to glycine betaine under salinity and hypoxic conditions. But obviously, functional studies would need to be done. So we don't know whether what we're seeing in glycine betaine responses under heat stress is telling us of a possible replacement of glycine betaine with yet another osmoprotectant, including beta-alanine betaine or even sugars. For that matter, we are not sure. So it's something that we would need to look into further. But at least it gave us, the review also gave us, you know, something to think about on our glycine betaine. So depending on what you're measuring and your stress system, you may not get what you thought you would get. In conclusion, I'd like to say we've got a lot to learn still, whether we are looking at individual stresses or a combination of these stresses in whatever system technique we are looking at, whether it's physiology, any of the omics technologies, whatever. There's still quite a lot to learn. But I also don't want to forget the root phenotyping studies, but this should be done in systems that allow unrestricted root growth so that we do not identify some external factors that would affect the results. You know, we are growing our plants in small pots, really. Even we still haven't gone into the field, which is something that we need to do. And regarding the osmolytes, I think we need to dig deeper and understand the different ranges of osmolytes that are produced and accumulate in sorghum under a range of these stress factors. So as to, you know, catalog really what is happening whether it's tissue specific or cell line specific or whatever. We still need to learn a bit more there, because much of the knowledge we have is mainly from Arabidopsis and maize and others. Just to end, I would like to say my many thanks to the University of the Free State, my postgraduate students who've done most of the work, both past and present, collaborators who have been very, you know, patient with me, and funding bodies who have assisted with the funding for these projects. And I thank you for your attention.
Thank you very much, Dr. Ankara, for your groundbreaking molecular study in plant stress responses and the proteomic work. I think it was very enlightening. Now we move on to the question and answer session. I think we only have 3 minutes for that. So the first question in the chat box is from Prof. Jacques Barna. He's asking whether will the SAA1441 variety also have a higher tolerance to drought stress. So I think it's pinning on your studies to say, from the work that you have done, is there any intervention or any approach that maybe could be used to make sure that maybe the drought susceptible variety can also probably have tolerance?
Okay, the question that I'm reading says SA1441. SA1441 is already the drought tolerant.
Okay.
Are you talking about heat stress? Are they asking whether or not SA1441 has also higher tolerance to heat stress?
Let's see. It says, will SAA411 also have high tolerance to drought stress? Does it already have high tolerance?
Yes, it already has drought stress. It's the one that I was showing you, you know, positive results in everything.
Probably, maybe if there was a swap in the questioning, maybe it could. I actually question on that as well, to say, would the one that is susceptible, did you get any insight or some information maybe on specific proteins that could be manipulated to also make it tolerant like the other one? Probably.
Oh, okay. All right. Thank you. Yes, I think, but we also got those ideas surely after doing the review. So what we found out that from the other sorghum review, the reviews of other studies, you know, these varieties, even if you're looking at them under control conditions, tolerant varieties of sorghum already have a high level of ROS detoxification enzymes or transcripts. They're already there constitutively at a higher level. Even the signaling proteins at a higher level. So I don't know if it means that having these already there, ready to just switch on after a particular stress has been imposed is important. I'm beginning to think what you have initially before the stress actually gives you some pre-protection. So definitely, if you are looking at I don't know if I could be able to go back to my maybe I won't share it, but if you're looking at the stress responsive proteins, which is basically what we also saw in the transcriptome, the upregulation of transcription related proteins, protein synthesis, protection of proteolysis, ROS detoxification and signaling processes. You know, all these classes of proteins are sort of priming the tolerant variety to increase a pool of more regulatory proteins, whether it's gene regulation or whatever, stress responsive proteins, which are going to protect the cellular components and so on. So maybe classes of proteins from those groups of those functional groups would be of importance, you know? And because also SA1441 is synthesizing proteins, but it is protecting them from degradation by inhibiting proteolysis. And I think that is sort of a well-crafted system in the tolerant varieties. But what I also wanted to speak to you about the heat tolerance of those two varieties, you know, I just had a picture there. It seems that SA1441, which is drought tolerant, also is heat tolerant. But that story is still in patches. I won't be able to give a good story to speak to you about those results at the moment.
Okay.
But yes.
Thank you very much. The last question is from Prof. Dave Berger. Do you have any plans in the future to perhaps make process between your two varieties, the susceptible one and the tolerant one, so that you can maybe at least explore into the genetic basis of what causes them to say one is on this extreme and the other one on the other extreme?
Thank you very much, Prof. Dave, for that comment. I think it's something that we should look into. I think I would need collaborators there, maybe speak to people who have knowledge on that and look into that. On the other hand, we're also looking at if, adding ABA in ICSB 338 could make some slight changes in its tolerance. But once again, it's still a project at its infancy, but there's still quite a lot to be done. And I think also taking these in the field and doing a field drought stress study would actually be meaningful as well, because we're still working on them at seedling stage. Who knows what happens later on?
No, thank you very much. I've got a comment from there's Nomatile Nombeu, who's saying, great talk to you, Dr. Ngara. Thank you very much, Nombeu. So to close, I would like to thank you, Dr. Ngara, for a great talk. It was quite entertaining and educative, and we hope you get more collaborators. And we also wish you the best in your current work and your future plans and future planned work as well. So thank you very much for availing yourself to this conference and giving this talk to us. So we welcome you to be with us for the final day today and until we finish, and we hope to work with you again in the future. Thank you very much. And
Thank you, Prof. Thank you.
Okay. Now, okay. Thank you very much, colleagues. We just got our plenary lecture now from Dr. Ngara. So we are trying to manage our time. I think we have about 10 minutes or 11 minutes comfort break, so that we will start at 10 o'clock, where we will break into a parallel session. So today we have only one parallel session. The first one is on ethnobotany, which will be chaired by Prof. B.E. Van Wyk. And then the other one will be plant taxonomy, plant schematics, and plant anatomy, which will be chaired by Dr. Lise Jobert. After lunch, we will now come to a single session, which will be on ethnobotany and chaired by Dr. Nobile Masonto. Then after that, I think later in the afternoon, there is also a SAAB annual general meeting, which will be running. Then we'll come back to the last part of the conference, which is the prize giving. So that one will start at 4:40. So I actually wish you all the best for the final day, and we hope you enjoy the last day of our conference as we finish off. Thank you very much, and all the best for the day. Thank you.